Hello students. So today through this video I will be discussing about the Mendelian inheritance, and mainly through this video I will be discussing about the Mendelian monohybrid experiment. So let's begin. Mendelian monohybrid experiment, it is also called Mendelian monohybrid cross. Monohybrid cross means cross between one pair of contrasting character. Mendel in his experiment, he mainly taken pea plant for his inheritance experiment. So here if we take one example of pea plant, and with the help of this example we will illustrate the Mendelian monohybrid experiment. So tall pea plant crossed with a dwarf pea plant. Okay, and tall, this is the dominant trait. Okay, so here we mainly discussing about one character, and that particular character is height. So in the height, two main traits are tall and dwarf. And tall, this is the dominant trait, and dwarf, it is the recessive trait. So as tall is the dominant trait, we have to use capital T for tall trait. Capital T for tall trait. And dwarf, this is the recessive trait. So we have to use small t. Now next question is, why we are just taking two capital T or two small t? Why not one? Why not three or four? Because pea plant, this is a diploid organism. Okay, that means they have two set of chromosome, and in that two set of chromosome, in their corresponding area, in a particular locus, the same kind of genes are present, which is helping in the same kind of characters, which is helping in the expression of same kind of character. So here we can see capital T. That means, suppose these are chromosomes. These are homologous pair of chromosomes. So here in this particular position, the gene capital T is present. Okay, that means in this homologous pair, in that corresponding area, in the same locus, capital T is again there. That means this particular portion of the gene of this homologous pair, they are controlling the character of height, and that particular trait is tall. And in the same way, here for two homologous chromosome, and suppose in this particular position small t is present here. That means here in the corresponding area of the same locus, small t is present. So this particular portion we just call them as allele. Okay, so here it is allele, here it is allele. So here allele for tall trait, and here small t allele for dwarf trait. Okay, so now we have to segregate the gametes. So we can segregate capital T, and here small t. So I'm just removing this particular portion. We don't require this particular portion here. Okay. So here this part is indicating the parents. This part is indicating the parental information. That means this is the phenotypic information about the parent, and this is the genotypic information about the parent. Okay, and here we actually segregate the gametes. As you all know, during the time of gamete formation, half number of chromosomes are mainly transmitting to their gametes. So here capital T from this diploid form, capital T from this diploid form, suppose single form, that means single set of chromosome is mainly transporting or transferring to their gametes. Okay, So that's why I have written capital T here, and I have to segregate this. So I have segregated that capital T. And in the same way, I have segregated small t and small t. So these are indicating the gametes. These are indicating the gametes. And now we need to make a Punnett square. We need to make a Punnett square. Let's erase this part. So we need to make the Punnett square. And in this Punnett square, we need to form the Punnett square in this way. And here we have to place the female gametes, and here we have to place the male gamete. So here suppose you take capital T. So these two gametes I have taken in this Punnett square in this part. And this small t, so these gametes I am placing here in this y-axis part. Okay. So suppose these are female gametes and these are male gametes. No problem if you take small t here and capital T here, there is no problem. Okay. Now if we make cross there, capital T or small t, capital T, what do you get? Capital T small t. Here capital T small t. Here capital T small t, and here same way capital T small t. So these are indicating the genotype of F1 generation. Genotype of F1 generation. Okay, and here if we see capital T small t, this is the genotype. And what would be the phenotype? The phenotype is tall. Here also tall. Here also tall. And here also tall. That means in F1 generation, Mendel, through his Mendelian cross or Mendelian through his monohybrid experiment, Mendel he observed that in the F1 generation, the offspring which mainly obtained here all are tall, and the genotype is capital T small t. Okay. Now what actually he did in the next part? So let's see. So these are capital T small t, and these are the genotype of the offspring. Same kind of genotypes are there, and same kind of phenotypes, tallness, are mainly developed in the F1 generation. Now next step. Okay. We have to remember one thing, that in the F1 generation we got capital T small t, the genotype of the F1 offspring, capital T is small t, and the phenotype of the offspring is tall. Okay. Now we have to make a selfing. What actually Mendel did in the next part? He did the selfing. Selfing means the self-pollination between the offspring, which mainly appeared in the F1 generation. So here I am writing selfing. Selfing between the offspring of F1 generation. Okay, F1 cross F1, that means the offspring which mainly obtained in F1. So we are making cross in between the offspring of the F1 generation. So what we get? Capital T small t. We got this kind of genotype of F1 offspring. So capital T small t is crossed with capital T small t. Okay. So now they are the parents. Okay, they will make cross. So here we need to segregate the gametes, capital T and small t. So these are gametes. Here also capital T small t. So now they are the gametes. So these are the gametes now. These are the gametes. These are the gametes. Gametes of F1 offspring. Okay, in F1 generation we got the few progenies, and now we have to place them. We are just making a self crossing or selfing, and here we have taken F1 into F1 cross F1. So in F1 we got capital T small t cross, and in F1 again we got capital T small t. So same kind of offsprings we obtain, and here we are just making cross, and we have to segregate the gametes. Okay, so we have segregated the gametes here, and I have written gametes. So now we have to make the Punnett square. Take the Punnett square. Okay, and here suppose I am taking these two gametes, I am placing them here, capital T and small t here, In the same way, capital T, small t I have placed here. Okay, so these two gametes I have placed in this position, and these two gametes I have placed in this position. Okay, so no problem. And now we just make cross. So capital T, okay, capital T, small t, then capital T, small t, and then small t, okay, small t. Okay, now capital T, it is indicating tall. It is indicating tall because capital T, it is for the dominant trait tall, and capital T, small t, here it is also again tall because small t is dwarf and it is the recessive trait. Okay, and dominant trait means it is always suppressing the feature expression of the recessive trait. So that's why capital T, as it is present, the small t it cannot express because capital T is the super ruling gene, super ruling trait here. Okay, so that's why phenotypically we are getting tall. Here also capital T, small t, tall. But here if you observe small t, here you observe small t. So small t, that means capital T is absent here, that means dominant trait is absent. So now the recessive trait can express. So small t is indicating the dwarf. Okay, so it is indicating the dwarf. And these are the offspring which mainly obtained in F2 generation. So these are the progenies which mainly obtained in F2 generation. And now if we observe the phenotypic and genotypic ratios of F2 generation, you can observe now. Suppose I am writing here the phenotypic ratio of F2 because I am observing here the F2. Okay, so phenotypic ratio of F2. Phenotypic ratio means the visible expression, the visible characters. So what are the visible characters here? Tall, no, tall is to dwarf. So now we have to count how many tall we get actually. So tall 1, 2, 3. So 3 tall, and dwarf 1. So 3:1. So this is the phenotypic ratio of F2 generation which mainly obtained in Mendelian monohybrid cross. Now we have to check the genotypic ratio. Genotypic ratio of F2. So genotypic ratio means the gene expression which is not visible. Okay, so we have to write this alphabet. So these are indicating the genotypes. So capital T. So if I write here capital T is to capital T small t is to small t. Now we have to see their frequency. Okay, so capital T if we count here, so how many times we are getting capital T here? 1 is to capital T small t. How many times? 1, 2 times, and small t only 1 time. So what we will get finally? We will get the ratio 1:2:1. So this is the genotypic ratio of F2 generation, and 3:1. This is the phenotypic ratio of F2 generation. So this is about the Mendelian monohybrid cross. Okay, I have taken one example here. I have taken one important character, that is height of the pea plant, and two contrasting traits actually I have taken, and this is not taken by me but this is taken by Mendel. Okay, and I have just following that particular method. Okay, and here I have explained this Mendelian monohybrid experiment by taking tall and dwarf traits. So here tall it was the dominant trait and dwarf it was the recessive trait, and here actually I have taken one important character, that is height. So one pair of contrasting character I have taken and I have made a cross there. So it is called monohybrid cross, which was done by Mendel, and through this experiment Mendel actually explained about the Mendelian monohybrid cross. Okay, so thank you for watching this video.